Okay, good. Thank you very much, everybody. Thank you, Caroline. So I'm going to give a short presentation on the application of free prediction models for drug discovery. I'm going to start with a disclaimer. So Caroline and Alex and I have talked about this, but there are definitely lots of opinions on this topic. These are some of ours. I would very much welcome yours in a discussion at the end. So as Caroline said, we can have a discussion at the end, and please type some questions as they occur to you into the questions box as we go through. So what I'm going to cover is just outline the scope of this presentation, talk about some sources of in silico models a bit, and then really focus quite a bit on assessing model quality in particularly the optimization phase, and then talk a bit about assessing model quality in general, and then move on to talk about HTS triage as a somewhat separate case, distinct from optimization. A few slides on logP and tautomers, and some things to conclude. So in terms of scope, we're mainly thinking today about models of pharmacokinetics and toxicity, not, for example, so much about activity models, although some of the principles will apply to those. And also, we're not really talking about docking and scoring in ligand binding sites, although again, some of the principles would be applicable. We have not attempted to put together a completely exhaustive list of all of the potential models and tools that might be available. We have got some, but please don't assume that just because it's not on the list, we don't think it's valuable. This is just a small selection. So in terms of the models that we are talking about, I'm going to touch a little bit on some of the kind of specific models, things like solubility, metabolism, absorption, and also some more general models. I guess logP isn't general in some senses, in as much as there's a very specific measured property that you're trying to predict, actually, but because it applies to lots of aspects of drug discovery, I guess it's used in a general way, and then some things like drug likeness and pKa. So we'll touch on both of those kinds of things. And I guess I wanted to start by just setting in silico models in a little bit of context in terms of drug discovery, which is that obviously in drug discovery, we use a whole number of models in the broadest sense, and many of them are experimentally derived, which you might term surrogate endpoints, that we hope are going to correlate with something useful, usually in human. So, you know, things like cytotoxicity and measures of in vitro metabolism are, of course, in themselves models of things that we think will be relevant to somebody taking a pill or an injection. And of course, some of the models that the in silico models that we put together are predictions of those surrogate endpoints. So just, you know, a little bit of context about some of the in silico models that we're talking about. So moving on to think about where you could go for some of these models. As I mentioned, this isn't an exhaustive list. I've listed these sources partly because we have touched on some of them in these sessions before. So Data Warrior, you will know, is freely available from Actelion, and the website. Ooh, I'm getting lots of beeping on my line. I hope nothing bad is happening. Caroline, if anything disastrous happens, I hope you can let me know.
That sounds fine at this end, Mark. No beeping.
Okay, fine. So Data Warrior is free to use, and then Swiss ADME you heard from last month, and there are lots of models available through that very useful website. Obviously, there is a slight distinction in that for Data Warrior, you can use it on your laptop, and depending on what you do, nothing you do will move outside your environment. SwissADME is on a website, and we'll touch on that difference a bit later on. ChemBench is another website offering very large numbers of models, and StarDrop is generally not free, but I've listed it here because many of you, I think, will be on the line, will have had access to StarDrop funded for you. So I wanted to, point out that that's also another good source of models. So there are many references at the bottom of the pages. Quite often I've included the full link so people can hopefully access them if they're just viewing the video, and the slides will be available as well. So some sources of models. Last month we saw a picture of this gentleman, George Box, and he came up with this often quoted saying, essentially all models are wrong, but some are useful. And of course, what I want to talk about to some length is how you might go about assessing how good the model is for you. And really, I haven't focused here on model accuracy overall. So I guess I'm not going to sort of say, look, we've cracked it, we've found the perfect metabolic stability model. More going to focus on how you might assess whether a particular metabolic stability model is useful for your set of compounds. And the other thing is, this is not intended as a session for people that might be building models and thinking about how to go about that. There are some very good guidelines available. One of the ones I like is this reference from Jamie and Kim in 2014. And I do think that if more people who are publishing models adhered to the principles in there, there would be better quality models available. Anyway, in terms of the focus of this presentation, then it's going to be on which compounds, assessing the models on your compounds, principally. I didn't want to dive straight into that before mentioning this paper, which I think is really interesting and valuable. So Jack Scannell presented actually at the UK QSAR a couple of months ago these ideas, and the reference to the paper, which is freely available, is down there. And one of the things that struck me about it is this quote that they theorized that an improvement in R, so an improvement in predictive validity, as they sort of operationalize that as R, of just 0.1, which doesn't seem like a massive amount perhaps, after doing a lot of theoretical modeling, they conclude that it is worth more than a 10-fold increase in throughput of your experiments. And this is all in the context of analyzing the ills of the pharma industry in the last 20 years or so. And one of the things they look at is the contrast between discovery of antibiotics when there were relatively small numbers of compounds available to look at, but nevertheless the end goal was achieved, and more recent times when there are millions of compounds to look at, but it is seemingly proving tougher than ever to discover new drugs. And they put some of that down to the use of quite a lot of models, not all of which have an R that is close to 1. Anyway, that is perhaps a slight aside, but I think is an interesting backdrop to thinking about model quality in general. So moving on to think about property prediction within a series. So the context here is, as I mentioned, optimization predominantly, and the idea really is that for the most part, it is going to be extremely valuable to have some experimental measurement of the properties you want to predict for your series, because that will give you a good way to understand whether a particular model that you want to use is working well or not. So the ideal scenario presented in this workflow is to be on the right-hand side with compounds from within a series where you've got experimental data and you can compare the model's predictions with the actual values that you've got. And you can carry out some simple statistical tests to look at that, and if you've got a good correlation and your unknowns seem to be are relatively similar in structure to those that are predicted well, then great, you're in reasonably confident area where you should be able to apply models. Of course, that's not always the case. We're going to talk about that too, but that is certainly the ideal scenario. So that's kind of over on the right-hand side. In the middle, we've got the area where you haven't really got very many experimental properties, but you have got some. And so in this area, you're really not quite at the area where you can say you've got enough evidence for statistical rigor. But of course, if the compounds that you've measured are in agreement with prediction, that's certainly better than them disagreeing. So that's a good start. On the left-hand side, if you've got no experimental values at all for the properties you're interested in, then it's going to be somewhat more risky. Ideally, you'd want to measure some compounds. You could look for several different models predicting the same endpoint and see to what extent you get consensus between those models. Here, you're going to have to do some work to try to understand, well, are the models built in the same way or differently? Because if they're all built off the same data set in the same way, actually having multiple models probably won't help very much because they'll be in alignment anyway. So really, you want models that have been built perhaps on different compounds or perhaps with fundamentally different ways of constructing models. So you'd need to do some digging there. You could also look for through databases like Kemble for compounds that are similar to the ones that you've got, which have got experimental values, and of course look for alignment between the model and those compounds that have got experimental values. You perhaps need to be a little bit cautious or aware at least that those models, if they're in the public domain, they may have used some of that data to train those models. And if that's the case, depending on how the model's been put together, that may give you an artificially warm glow that the models are predictive, when in fact it's more true to say that they are predictive because they were trained on that data. All of this is a sort of another dimension to think about, which is to do with how well that endpoint is generally predicted. So for example, you'll see an example of plasma protein binding, which is probably not as difficult to predict, for example, as microsomal or any other type of clearance. So if your endpoint is very difficult to predict, then you really need to have some measured values to compare with the prediction, and ideally probably more than 6, as we'll see in a minute. If the endpoint is perhaps a bit easier to predict and more generally predicted well across a range of compounds, then it's probably going to be able to get away with fewer measured examples. And on the left-hand side, some of those where logP is a big component are probably types of models where you are, it may, the models may work more generally, and so those are the kinds of things where you may be able to get away with fewer endpoints. One of the things that is kind of underlying this is this idea of property prediction within a series. So a question might arise, well, what's a series? So for this purpose, really, it's kind of what a medicinal chemist thinks about as a series. So the sort of cartoon representation of chemical space here, the idea is that the green compounds are predicted well by a model, and the blue compounds are similar enough in structure that they're likely to be predicted well by the same model. And that's the sort of nice scenario that we were talking about earlier. Of course, if the compounds are quite different, then you know, or to some extent, all bets are off. You can be less sure about whether the prediction is going to be good or not. In terms of what's a series question, so as I said, really it's the kind of thing that a medicinal chemist would call a series, generally speaking. So for example, compounds with the same core and broadly similar R groups with perhaps the same sort of charge state. There are more mathematical ways to look at this, and this is a paper from a few years ago from the AMG group, who looked at a variety of different similarity methods and coined this idea of using belief theory. And from my experience, then these kinds of the idea of what's a series is more or less coincides with this sort of halfway up this curve, roughly, of the percent likelihood of similar percent activity. So the sort of 20 to 30% area. So in these commonly used fingerprints, ECFP4, for example, it's going to be in the sort of 0.5, 0.6 range, roughly speaking, in my experience. So no hard and fast rules, but as a sort of overall guideline. Again, another thing that's just worth a couple of seconds is just to think about when to consider a log plot or understand a log analysis of your endpoints. A lot of times I still see graphs of concentration which isn't logged, and I think generally speaking it really ought to be. I'm not going to go into all of the reasons for that. I think most people probably appreciate them. But PKIs, PIC50s is one sort of treatment, or just taking a log. Of course, things that already have logs within them, things like logPs, logDs, they don't need it. And time is also usually not logged. So just to move on to think about some of the statistical tests. So we were talking about this idea that you've measured some compounds for some of the examples in your series, and you want to know whether this model is working well. So how do you go about checking that out? And I'm going to mention three different approaches. This is by no means all of the things that could be done, but it's a useful start. So if you get good statistics from these kinds of approaches, and your compounds are similar to the well-predicted model, the well-predicted compounds, then your model is likely to be useful. So the first two are useful with predictions which are numbers, and the third one is to do with classification. So as an example, to look at some of these methods, I'm going to start with data from the Solubility challenge. So Jonathan Goodman, October last year, spoke about the Solubility challenge and solubility in general in one of these sessions. So the link to that YouTube video is there, and there's the data available from his website. And if you download that and put it into Data Warrior, so on the y-axis here, we've got the log of kinetic solubility for the compounds in the data set, and on the x-axis, predicted solubility, predicted log of solubility. So this is a scatter plot of the prediction against actual, and what you can do is look at the size of the error. So it's easiest to think in terms of unsigned error for some purposes. So simply just square root with the error squared, or there's other ways to do it, but that's simple. In Data Warrior, if you haven't come across this, I'm going to refer to Data Warrior a few times and how you could go about actually doing some of these things. So that would be the data menu item, and then add calculated values, and you basically put in the equation that you want to in the kind of way that you might do in Excel. There's some help within that menu. So if you do that to this data and then plot, you calculate some bins. These are binned in 0.5 units. You can see that the mean unsigned error, so the average of the error, is about 0.8, and the median is 0.62. Actually, it's not terribly easy in data warrior to get a hold of those, but they are available if you plot box and whisker plots, and the red line is the mean and the black line is the median. You can also see from this bar chart that about 91% of the error is essentially within tenfold of prediction. And then there are some significant outliers. So that just gives you a kind of overall picture of the error in the model for this data set. Other types of plots looking at error. So a useful thing to do sometimes is to look at error and plot it either against the experimental values or the predicted values. And you can see here, if you plot it against the experimental values, that the size of the error looks to be potentially larger at the extremes of solubility. So at very high solubility on the right-hand side here, or very low solubility, the model seems to differ more, at least have the potential to differ more than it does perhaps in these more middle ranges. If you take away the sign and just so in other words add back in the sign of the error, you can see that there is an apparent bias as well in the experimental solubility towards a positive error at high solubilities and a negative error at low solubilities, which interestingly isn't the case for the prediction itself. So the predicted value is pretty much has a mean of 0 pretty much across the piece. That's for the signed error. Obviously the signed error is going to have a mean of about 0 if there's as much overestimation as there is underestimation. So that's looking at error. Correlation is another thing you might want to look at. So I'm talking here about the Pearson correlation coefficient, and there are two symbols here. R is technically for the sample, and rho is for a population. So essentially the similar things, different technically to statisticians, and it's a measure of association between two variables. So it takes the value +1 or -1, between +1 and -1, where +1 and -1 are straight lines, as you can see in the bottom portion here. A couple of words of caution. So if you've got only 2 points, you will get +1 or -1, which might look good, but with 2 points, it's probably not necessarily useful. It does assume that the underlying relationship between the two variables you're looking at is linear. So you can see on the very bottom that somebody's decided to graph up some pretty patterns, which are clearly not a random relationship for the most part between variables, but for which you still get a correlation coefficient of 0. And also it doesn't imply that y equals x. So if you can see in the middle set here that the slopes vary enormously, but they can still be exactly 1 or -1 in terms of the correlation coefficient. Okay, so just going back to the solubility data, the data warrior solubility predictor against the kinetic solubility measurement, you can see that the Pearson correlation coefficient comes out at 0.716. For a different solubility prediction, it comes out higher, 0.83. And this is one that's available through Swiss ADME. So these were all put through the Swiss ADME website. And that's a reference for that solubility model, and that's all available off the Swiss ADME website. So okay, so it looks like it's a larger number, and the larger the number, whether it's closer to 1 or closer to -1, indicates that it's closer to a straight line, basically, and that's great. But is it statistically significant? So just because you have a number that looks like it's quite high, there's also a question about, could that distribution have arisen by chance? Okay, and then there's another question you want to ask as well, which might be, well, it might be statistically significant, but is it useful? So for that, we're just going to take a look at a different set of experimental data and modeling. So this is looking at plasma protein binding. So this is a slide from a presentation on the PK tool. So why, obviously, just a bit of background, if you're not familiar with it, the drugs frequently bind to human serum albumin, and that gives you bound drug, and under the free drug hypothesis, it's only the compound that is unbound that can do things like move across membranes, bind target, and be cleared. So it's a very useful concept in thinking about drug discovery in general and metabolism particularly. And the fraction unbound, which is kind of the flip side of protein binding, that's going to vary from 1, which is completely unbound, to 0, all bound. In the PK tool, just a very brief reminder of that here in the video here, and some intro and links you can go and find online. We've talked about this before. You need to know basically the fraction unbound to calculate the free drug from a given human dose, or to calculate a human dose. So that's one of the reasons why you might be interested in calculating it. So thinking about this is from a project that I've been involved in, and this is a plot on the y-axis of the free fraction in mouse plasma against logP. 2 different measures of logP calculation, the one that's in Data Warrior and one that's available from ScienceCloud. And the reason we have that is because that's where the data in this project is stored. So one thing to note is that there seems to be quite a good straight line for both of them, but there are a couple of compounds up here which are lying off the straight line, and very significantly so here. And you can see that these are the 2 same compounds, very differently predicted by the Data Warrior C-logP and ScienceCloud S-logP to the extent of about more than 4 units. That's pretty unusual. These are somewhat unusual compounds. It contains a polar prodrug group, so it's that's being handled very differently. For the sake of this analysis, and actually really because this is a bit of a special case, we've just taken those 2 compounds away for the next plot, and that changes the statistics a bit, and you can see that really they're quite nice straight lines, these. So just to look at a bit more detail on this question about whether or not this distribution could have arisen by chance, I suspect most of you looking at it would think, well, no, that looks like a pretty unrandom correlation to me, and that's the way we're going to find it. But just to show the maths that goes into that, firstly, if you want to calculate the correlation coefficient yourself, you can do it, as you can see in Data Warrior. You can just right click on any scatter plot and ask to show that from the statistics. But in Excel, you can also calculate it, and you just give the correl function these 2 arrays of the 2 different numbers, and it'll calculate the same thing for you. I won't go into the maths. You can obviously Google that if you want. So that's calculating the correlation coefficient itself. Then what about how likely is it that that could have arisen by chance? So for that, we need a test of significance, and there's 2 steps to that. There's this formula to calculate the t-value, and n is the number of observations, which is 23 in this plot, and then r is the correlation coefficient. So you pops a number from that equation, and you use a t-test table to determine how significant an r of 0.9 is in that with that many samples. And in Excel, you can do that calculation. So essentially, it looks up this t-test table for you, and it tells you that in this case, as I think you would all agree, this is very significant. So p is much less than, essentially, it's not random. So the correlation is highly significant, and S-log-P can be used to estimate the free fraction. For analogues, but probably not prodrugs, at least not without a bit further work. And that actually turns out to be particularly useful at very low free fractions if you're interested in very low free fraction compounds, because it's experimentally very hard to measure low free fractions. So all of this calculation might be a bit of a bore. I have produced a table which hopefully will make things a bit simpler and save some maths of R versus N and statistical significance. So the values are the p-value, and loosely that's the probability that the R value might be observed by chance. So where you've got very low values, then you can be pretty confident that that didn't arise by chance, and there is a real correlation between the two things that you're plotting. So these values are all, these are the p-values. So you can see the green ones are all less than essentially 1%. The orange values are all less than 10%. And the red values, you really probably should be fairly suspicious about whether or not that's a genuine correlation. And one of the things you can see is that at a low number of observations, 3, 4, or 5, well, with 3, you've got pretty much no chance of having a highly statistically significant correlation, even if you've got a straight line. And you can see that the right-hand side of the graph here goes from 10 and then jumps to sort of 20, 50, 100. At high numbers of observations, you can actually get statistical significance with very low R values, and we'll have a look at that in a minute. Just a word on what's acceptable. There isn't really a right answer for that, although 95%, so p less than 0.05 is often used. It does depend a little bit on the context. So for example, if you're testing a single model, which you're fairly sure is the one that you want to check, then you can probably consider a number closer to 0.05 or even 0.1, depending on the circumstances. If, however, you're randomly testing the association between lots of parameters, it's possible to calculate very large numbers of parameters for compounds, and you're randomly looking to see if any of those are associated with your experimental endpoint, you're going to need a very high confidence, because if you have a lot of random numbers, there's a good chance something's going to correlate with your real endpoint. So another example. This is showing a correlation between a calculated property on the x-axis and an experimental property on the y-axis, and it looks like a reasonable straight line, and we've got 0.756. It's minus because the slope is down here. And also, just if, in the way that you like to think about using these kinds of things, if you have a cutoff of 0.6 in this calculated property, you can see that it splits the 10 experimental values into 2 groups quite nicely, those with low experimental values and those with high experimental values. So that all looks nice. And looking at the statistics, you can calculate the p-value, and it comes out less than 1%. So that looks like this is a nice model. And if it is an example of just one calculated property and you're looking to see whether it predicts your experimental, I would agree. I think this does look like a nice model. In terms of how this was derived, a slight warning, I guess, that this was actually 2 sets of 10 random numbers. It wasn't any 2 sets of 10 random numbers. I just generated 50 sets of pairs of 10 random numbers and looked for that set of 10 pairs of random numbers which were had a high R value. So the next best sets are those that are below. So the point is, it was this point that I made earlier. If you're testing something that you're fairly confident is worth testing, from a small number of predicted experimental endpoint, then perhaps a p-value that's not too high is okay. But if you're testing from a large number of random properties of your compound to see if any of them happen to predict for your experimental endpoint, then you really do need to be cautious about over-interpreting what looks like a reasonable correlation. In terms of getting hold of some of these correlations, Data Warrior I mentioned calculates R for you. But it doesn't calculate significance. So you can, in fact, get a complete correlation matrix for all the data that's in Data Warrior from this menu item. And this is the kind of thing that you'll get. and so you can see which properties correlate closely together, or for a particular scatter plot, just right-click and then show the statistical view options. So that's two ways of looking at statistical analysis of real numbers. The last one I was going to show is what's called the confusion matrix. So this is useful to examine the predictivity of classification models. So you've probably seen the kind of thing before. You've got actual value divided into positive, negative, and predictive values divided into positive and negative. So for this, I've taken a look at the rule of 5 and a set of oral and parenteral drugs. So I've used the data set that's available in Data Warrior of approved drugs 2015. So if you go File, Open Reference File, there it is. I've then filtered it just to select 847 compounds for which the route of administration is known and also is either oral or parenteral or some combination, because some compounds obviously can be used for both. I've then used Data Warrior to calculate the rule of 5 properties. I haven't gone into any analysis of whether logP5 in Data Warrior's logP implementation is the right value. This is just really for demonstration of the stats. It's not intended as guidance for designing oral or parenteral drugs. So if you then look at the confusion matrix, this is there are 847 compounds in total, as I said. There are 114 which are parenteral only, and 733 which are either oral only or oral and parenteral, and then there are 703 compounds which are rule of 5 compliant, 144 which fail at least one of those. And so armed with these numbers, you can calculate a whole bunch of different things if you want to. I've just gone for some simple things. So for example, 13% of the total set are parenteral only. If you look only at the rule of 5 fails, then 34% of that data set is parenteral only, and 9% of those that are rule of 5 passes. So there's about a 4-fold difference, close to a 4-fold difference in those two. And you can also do a significance test associated with this distribution and ask whether it's statistically significant or not. And again, if you've got fairly large numbers, it's relatively easy to get statistical significance. This is highly significant. That test, I'm not going to go into the maths of it, but it's based on looking at how would 847 compounds distribute themselves if you had a random distribution where you still had this kind of bias towards rule of 5 and oral, and this is the kind of distribution that you would get. So really the statistical test is asking the question, how different is what we see in the true case from what you would see from this expected random distribution? Okay, and with confusion matrices, there are a whole lot of lovely things you can do. Lots of other things you can calculate: positive predictive value, false discovery rate, false positive rate, true, all of that kind of stuff. The test of statistical significance I used is available here. You can calculate it longhand if you want to, but obviously everything's available online as well. Okay, so that's looking at confusion matrix. Just one word on deriving classification from numbers. So people quite often do this, particularly where the correlation doesn't look all that great. They say, okay, well, it's not a straight line, but if I take a cutoff at 3, then above that, things look really bad, and below that, things look quite good. And so I just wanted to sort of explain and look at that a little bit. So again, this is another experimental situation. This one is looking at hepatocytes, hepatocyte clearance as an estimate of in vivo clearance. So on the y-axis, we've got the in vivo clearance. On the x-axis, we've got hepatocyte clearance. The hepatocyte clearance has been scaled to the same units, liters per kilogram, and you can see it's a reasonable scatter plot. And in fact, with a correlation line like that with 0.77 with 37 observations, that's very statistically significant. Okay, and you can see that if you start to draw a cutoff at 10 on this, actually probably worth just going back and just pointing out that although it's quite a nice straight line, it doesn't go through the line of unity. So the point 1010 here and the point 11, so the straight line of unity would be up here somewhere. So it's offset quite a bit, but correlation coefficient doesn't worry about that. So the other thing to point out is if we draw a line at 10, then you can see that all of the a large proportion of the low clearance compounds in vivo, those with clearance below 1, also have mouse hepatocyte clearance below 10. And there's only one which has is a false negative, where we'd miss this one. Okay, so if we threw away everything with a mouse hepatocyte clearance above 10, we would lose this compound because we might never test it in vivo. Okay, so we can construct a truth table from this, or a confusion matrix from this if we want to, and it looks like this, and we can do a statistical test of that as well if you want to, and in this case it's highly significant again, not surprising. And this tells you that in the total data set, 43% of the compounds are low clearance in vivo if we define it as less than 1 liter per hour per kilogram. If we progress only those compounds with a mouse clearance of less than 10, then we would enrich those with a low clearance. So of those with this set here, 68% of them have an in vivo clearance less than 1, and would only miss one of the 16. So that looks quite nice, and it is quite nice. And with a decent R value, it's not surprising that it's quite nice. Just one sort of word of caution, I suppose, particularly where the correlations get slightly less good. So you've only got to move the cutoff a little bit. So instead of saying 10, we'd say, well, let's move it down a bit to 9, and this time we'll say we don't want to miss anything, which is an in vivo clearance below 1.5. And now the proportion of compounds in the total data set is 43% with low clearance, and we can enrich again quite significantly to 81% if we move the data set down. But now we miss 3 compounds. Okay, so one needs to be a little careful, because it's always tempting to be very sort of cute about where you draw the cutoff, and you need to be sensitive to the fact that really, is it justifiable to say that 9 is different from 10 in terms of the experimental measure? And if it makes a big difference to your conclusions and analysis, then I think you're in dangerous territory in moving the cutoff quite so finely. So I think at the very least it's worth looking at the scatter plot, even if you are going to turn it into a truth table type of analysis. Of course, if you're making decisions, and that's what this is all about, you may need to use cutoffs or use multi-parameter optimization, and we've spoken about that before in these sessions. But do think about the impact of changing the cutoffs just a little bit, and there are ways of looking at that with ROC curves and things, but I haven't gone into that here. So, summary so far. It's about an analysis for your compounds. I'm going to have to speed up a little bit, or we'll be here forever. So, a couple more things about model quality more generally. This is worth doing if you're interested in that scenario of comparing lots of potential more or less random predictors of your endpoint. There's a big risk of overfitting, as we've talked about, so looking for a very statistically significant R value is one thing to do. It's also worth trying this Y-scrambling idea, which basically just randomly sorts the true experimental values between the compounds, then completely start your analysis again and see if you get something popping up that looks as credible as your first analysis did. If you do, then probably your original model is probably worthless. Okay, so some stuff there and how you might go about that in Excel if you wanted to. Large data sets and statistical significance. I mentioned this earlier. This is the approved drug set, the full approved drug set. If you calculate molecular weight for this and correlate it with daily dose, you actually get a statistically significant correlation. But the the slope is almost 1, almost 0, and I'm sure that it's not a particularly useful correlation. So it might be statistically significant, but it doesn't mean that it's automatically useful. Dynamic range, this is just to point out that you may well find in small data sets in particular that you've got a narrow dynamic range in experiment and or prediction. So for this example, we've got mouse liver microsomal clearance varying between just less than 0.2 and just less than 0.8. It's only over a factor of 5. So the experimental variation is probably at least 2 to 3-fold, and the logP range is small too. So really it's not possible, I would argue, to draw any meaningful conclusion from this other than we need to measure a broader range of compounds before deciding whether there's a useful model to apply. So this is something Jim Metz communicated to me a while ago about the error landscape. So I will allow you to look at that in your own good time, in the interests of time. Applicability domain is about the response and chemical structure space in which the model makes predictions with a given reliability. So it really depends in large part on the training set and the descriptors. So if the model's been trained on a large diverse set, you can expect that it might be relevant to a reasonably large range of compounds. Just a note of caution, though, that just because it has a large applicability domain doesn't mean that you're going to get accurate predictions, only that it was developed with the intention of being predictive across a range of compounds. You still have to check that out for yourself. So we've talked about model application to optimization. I also want to touch on HTS. So in HTS, you probably have you're not going to have sufficient experimental properties most of the time to check whether or not a model's being predictive. So unless you've got a very specific need, I guess our feeling is that you're better off using phys-chem properties, and the rule of 5 is not a bad guide. This is a paper which I would recommend actually from the MMV group, looking at an HTS triage, and as one of the steps in their triage they used quite a generous cutoff of molecular weight 600 and xlogP of 6, and you can see some other things in there. So these simple properties do have at least some association with lots of things that are relevant to global health, but I think it's going to be quite hard to determine that specific models are really going to be very appropriate in HTS triage, or it's going to be quite difficult to determine whether they're giving you the right answer or not. So a simple way out really is to use phys-chem properties. One other note about this paper is that in the process, they also looked at known pharmacology of the compounds that they were triaging down, and we've talked about that before as well. And you can do that with a KNIME workflow, or indeed in Kemble, and there's a YouTube video you can look at about that. A couple of words on lipophilicity. You can see the difference there between logP and logD. Calculated methods for logP are obviously often used, and there are different ways of deriving that. There's a very nice discussion about that in last month's video at about 10 minutes, and each method can give different answers. And why do we think about lipophilicity? Well, I guess a lot of people have spoken about this over a long period of time. Here are some of the reviews. One of the very early ones, of course, Leo and Hansch, going through rule of 5, and Mike Hand's molecular obesity, Paul Leeson's analysis of different companies' approaches. Just to point out that different logP predictors can give very different answers. So at the very least, it's useful to be consistent in your analysis if you're using lipophilicity. just to wrap up with a couple of final thoughts. So the difference between using websites to make predictions in your own machine. So obviously, if you're using a website, you need to put structures in that other people, at least in principle, could see. So one possible risk mitigation, obviously you can look at the website to see what their policy is. You can also sometimes enter parts of a molecule that you're interested in, or molecules that are similar to but not the same as the one you're trying to predict. In some situations you get a choice. So Kemble, for example, you can search on their website. There are also ways they allow you to download their entire database. One example, Swiss ADME, they discuss the principles on which they could look at the structures. They are indeed stored, but they don't. So essentially you have to decide whether that's okay for you. And this is some of the output that the Swiss ADME approach generates. Very final word on tautomers. So we had a look at tautomers because obviously there are some situations in which tautomers can give quite different values, and logP is no different. The key message here really with tautomers is if you are consistent, then you should be okay, really. It's a different story for docking, where you need to get the right tautomer, really in thinking about properties like PhysChem and most of these models. It's probably more important to be consistent than to get the right one. Almost finally, in decision making, so medicinal chemists make decisions all the time. We have to. And the models may not be perfect, but neither are medicinal chemists, let's remember. And we did some work a little while ago looking at comparing some in silico models with human predictions. So this is an example from it where we looked at solubility, and we were asking the humans to bin the compounds into high, medium, and low solubility with particular criteria, because we didn't think it was fair to ask them to actually give a number. And so we got the models and the medicinal chemists to do the same thing. And you can see through this measure, which is a measure of a way of scoring a 3x3 contingency or truth table, that actually, although the models aren't perfect, they generally outperformed the humans. Molecular weight and logP, they have negative values, but actually you can flip those up the other way. So don't imagine that they were bad. They had values sort of around here, 0.3 to 0.4 range. Okay, so actually models weren't perfect, but in this situation they did outperform experienced medicinal chemists. So do assess model performance for your compounds, and hopefully, I think, so this goes back a little bit to the Scannell paper. My personal feeling, I guess I was sold on that, is that really the quality of decision making is very important and perhaps needs a little more attention than I have tended to give it over high throughput. And there are several sources of free models and some good tools to investigate whether or not they're predictive for your compounds or not. That's where I will stop. Thank you.
Great, thanks, Mark. Thanks for managing to cover quite such a huge volume of information there. If anybody has any questions, would they like to type them into the question box? I have one to be getting on with. This is from Chris Swain, who says...
Questions or opinions?
Questions or opinions, yes. Would you like to comment on local versus global models?
Gosh. Well, I think, Chris, maybe we should ask you to comment too. I guess I don't have a particular prejudice against one or the other. I think a lot depends on how each is being constructed in terms of what's going to be most applicable. But I'd love to hear your thoughts.
Would you like me to unmute him? Right. Chris, you're unmuted.
Hi, can you hear me then?
Yep.
Yep.
Okay. I think the global models are excellent when you're looking at trying to triage high throughput screening and things like this. But I think once you get down into the nitty-gritty of lead optimization, it's perhaps better to start thinking more about building your own local models.
Yeah, I suppose it depends a little bit on what you're putting into them, doesn't it? Because if you can put in phys-chem properties, then they're going to predict for quite a wide range of compounds. But if you're relying on fingerprints, for example, in a local model, then it would be still quite specific. Do you have any thoughts on that?
I think once you get down to, as I say, lead optimization, it's worth spending the time actually getting experimental values for things rather than relying on predicted or calculated properties.
Yes, yes, no, I agree. I think you've got to have experimental properties that determine whether or not something that you want to use as a model is going to be any good.
And also, one comment is that it seems to be very common that people do mix up measured log p values with calculated log p values. The two have become kind of interchangeable, whereas in actual fact they're not. Do we have any other questions? No? Comments? No? Well, okay. I think we're nearing the end of the hour, so I'll just leave a couple of seconds if anybody wants to ask something, and just say that there won't be a webinar next month because it looks like everybody's on holiday, judging by the number of bounces I got from emails this month, and indeed I am going on holiday. But we will be back in September with a talk from Antoine Dana and Vincent Zuete from the Swiss Institute for Bioinformatics, who talked about the Swiss ADME server in June. And this time they will be talking about structure-based design and some of the tools that the Swiss Institute make available for everybody to use. And hope to see you back then. So I think there are no more questions. So I think we'll say thank you to Mark for a great presentation and pulling all that material together. Look forward to seeing you all in September. Thanks, guys.
Thanks, Caroline.